All right, we'll move right along. So next we have Gregory Smith, and Gregory will be discussing the analysis of neuropulmonary access following lunar regolith simulant exposure.
All right, thank you so much. Let me share my screen. Okay, can you see this okay? Okay, so
yes, that looks great.
Okay, so my name is Greg. I'm a first-year master's student at Stony Brook University Department of Pharmacological Sciences. So the background and motivating force for this study was kind of to assess the toxicity effects of lunar dust to human health. So during the Apollo 17 mission, people came back, reported hay fever, reportedly smelled like gun smoke. Apollo 17 astronaut Jack Schmitt reported having his nose like inflamed, swollen after the exposure. So we were kind of looking to see what type of effects this can have on health. So the experimental overview: how does the lunar dust simulants affect health? Is it associated with disease? To answer this question, I took lunar dust simulant, the lunar Mars simulant, introduced it into mouse airways and compared it with saline and titanium dioxide controls, then took out brain and lung tissue and had them assessed for any pathological changes. So this is what lung slides look like underneath the microscope. So all these little airs, these little like sacs, these are your alveoli. And here we have an A is just saline treated. And then we can see these refractile crystals visualized under dark field microscopy. And in C, we could actually see some of the dust actually being engulfed by immune cells. So the dust was definitely in the airway. And from the mice with these dust in the airway, we were able to take out the brain. So in neurological tissue, the brain's immune cell is called the microglia. So it becomes activated and diseased and inflamed. So top row, we have saline treated, and we can see the blue is just the cell nuclei. The green are the microglia at a resting state. Treating with lunar Mars simulant, the microglia become branched, activated, appear to increase in number, inflamed. And with titanium dioxide or anatase, it's the similar effect. There has been one study reporting that titanium dioxide exposure is toxic to the central nervous system. So there appears to be morphological changes in the brain's immune cell following lunar Mars simulant exposure that is associated with neuroinflammation. I'd like to acknowledge the Circle Lab, McLean Lab, the Glotch Lab, and the RISE2 grant, and the survey community.
Thank you, Gregory. What a cool, different take on what we've been discussing today. That's really awesome.
Yeah, thank you.
All right.